So again, everybody, thank you for joining us for the Amnis Image Cytometry Data Analysis Advancements webinar. The presentation today will cover recent enhancements in functionality in IDEAS software with a focus on high throughput and batch analysis of data sets. This portion of the webinar will be presented by Brian Hall from Amnis, which is a part of Millipore Sigma. And we're also happy to announce on the Novo Software FCS Express side of things the new direct compatibility of Amnis and IDEAS.DAF files with FCS Express. Towards the end of the webinar, we'll be explaining and focusing on reporting and batch analysis of Amnis-derived datasets in FCS Express Image Cytometry, where you can link up the raw DAF and CIF files from your Amnis IDEAS software into FCS Express. Later on today, we'll also be hosting another webinar that will be focusing specifically on working with those data sets in FCS Express. So please take a moment if you haven't already to register for the second webinar. You can find that on the homepage at www.denovosoftware.com. So without further ado, I'd like to bring Brian Hall from Amnis on the line. We're going to make him the presenter now to begin the webinar. Speaking about some of the new functionality and the recent enhancements with IDEAS software. So Brian, I think we have you on the line. You can take it away.
Great, thanks a lot, Sean. I appreciate the introduction. And as Sean pointed out, we've been working with the Novo Software group and FCS Express to facilitate some of the batch reporting and make using the image stream much simpler. And especially when it comes to reporting your cytometry data. But we thought we would take this opportunity to go through some of the enhancements and some of the novel applications and new applications that we have coming out for the image stream. Some of the core advancements that have taken place over the last year or so with IDEAS are really allowing us to look at new applications. Some of the things such as the adaptive erode mask, component masking. We have new level set masks. There's now a watershed mask in IDEAS. We have one new feature, the bright detail co-localization three-way. We've enhanced some of the reporting tools, such as including the feature value with the imagery when you copy out your images, as well as changing the display order. So you can now order the images by a feature value, or you can order them by number. IDEAS enables up to 2,000 features, and our export tools have been enhanced where you can do multiple FCS data file export right from IDEAS to facilitate working in third-party software. We also now have histogram smoothing. So Amnis has continued to develop a number of applications. But when you look through the literature, the vast majority of new applications are being developed by our customers, which is exactly where we want to be. So we're continuing to get a really nice publication history, and been some really fantastic work out there that have brought to light some new applications. What I'd like to take this opportunity to do is go through and talk about some of the new applications that we've been developing in-house and that our customers have been developing. Primarily, we've been using some of these new masks that we've enabled in IDEAS to look at a lot of different bright field morphologies, and that includes looking at sickling and red blood cells, analyzing sperm cells, also looking at algae morphology, just using the bright field information. We've been developing algorithms for AML and ALL. There's been a number of good publications about microparticle analysis. We're also trying to enhance more applications in clinical diagnostics. So even though we're a research platform, we're still developing research clinical diagnostic applications, and we're hoping to get compliance on some of our clinical diagnostics over the next couple of years. We've released this year a protein aggregation kit, so you can now do a beta test of our protein aggregation kit, which allows you to fluorescently label protein aggregates and quantify the concentration. Along with microparticle analysis, we've also been looking at some exosome internalization. There's a number of good publications that have just come out about looking at micronuclei And of course, we still have a number of groups looking at circulating tumor cells. And then finally is our future goals and aims here at Amnis. Part of that is to look at multi-parametric analysis, which is doing things like taking high-dimensional data sets and reducing it into bivariate form for gating and for easy description of data sets that leverage hundreds, if not thousands, of morphology-based features. So those are some of the things we have going on, and that we hope to see more data coming out over the next year or so. So to get started in a description of how some of the new masking has been used, I like to use this example of just looking at a simple cell cycle profile. These are THP1 cells that were fluorescently labeled with DAPI. And in the top panel here, you can see we get a nice cell cycle profile looking at G0, G1, S phase, G2M. I should point out that FCS Express will now model the curves under this DNA profile so that we can look more specifically at these three major phases of the cell cycle. But as you know, with image cytometry, we have the ability to use nuclear morphology to characterize the mitotic phase. And when you look at the associated imagery, if we have round nuclei like this, and 4N DNA content, these are the G2 cells, and the mitotic cells have condensed chromatin structure. So you can see that these cells are preparing to divide. But using a multi-parametric approach where you use hundreds of features combined with some of our new masking, you can really go in and quantify the different phases of a cell cycle. So this is a quick comparison that shows our current masks along with the watershed masks. So you can see the default mask is M7. If you apply a intensity threshold to the masking at 90%, you can discriminate two chromosomal centers and anaphase cells, and the watershed mask also does a nice job of separating those two cells out. So leveraging these masks, we can then go through and look at the mitotic cells and characterize the different phases of mitosis. Here, telophase cells, using a telophase classifier, again, that uses multiple parameters to characterize this morphology. We get about 4% of the mitotic cells are in telophase. Removing the telophase cells and then focusing on the mitotic cells and not telophase cells, we can characterize the anaphase cells. And anaphase cells are characterized by two chromosomal centers with tubulin fibers connecting this to the two centers as the cell is dividing. Creating a third population of the mitotic and not telophase or anaphase. We can characterize prophase versus metaphase, and this is easily done. Prophase shows all of the chromosomes lined up on the chromosomal plate with the tubulin fibers attached to either pole, and then prophase cells have very condensed circular chromatin structure. So using a multi-parametric approach in some of our novel maskings, we've been able to enhance the characterization of up to 7 phases of the cell cycle. Now, one of the key enhancements that we've done lately is what we call component masking. And those of you who are familiar with ideas but maybe haven't taken the time to learn about component masking, I would encourage you to do so. It's a really powerful tool. And essentially, the tool came out of the need to characterize components within a single image. So if you look at this image down here at the bottom of your screen, this is a single cell, and it has 3 phagocytosed particles within it. One of them is at the membrane here. And the goal of this is to measure these particles independently, either based on size or intensity. And the way that this works in IDEA is you essentially go into the mask manager, select a component mask from the function manager, choose a parent mask with many components, and in this case, this initial mask here has 3 separate components. Next, we want to choose a ranking feature, and what this will do is rank these individual masklets based on whatever feature you choose here. And you can choose intensity. In this case, I've chosen area, so it's going to rank them from either in ascending or descending order, and here I've chosen ascending. So it's going to rank them in order of the smallest mask to the largest mask. And if you look at the imagery down here, you can see this is a 3-component mask. The area of the smallest bead is 20.25 microns square. Component mask 2 is going to mask the medium-sized particle, and component mask 3 is going to mask the largest particle. Now this requires you to iterate through the component masking 3 times to get these 3 individual masks. But once you have each of these particles characterized or identified, you can go in and quantify either the size of these particles or the total intensity. And so this has been a powerful tool that's going to allow us to do a number of new applications, including a more detailed phagocytosis. We can leverage component masking to look at fish spot intensity, fish spot quality, bacterial particles, intensities and areas, as well as looking at asymmetric cell division for immune cells. Another mask that was developed and recently implemented in ideas is the level set mask. And this shows you a series of red blood cell morphologies with the various level set masks. It kind of gives you an idea as to what you can expect from them. The level set bright is going to identify just the bright regions within the bright field image. The level set mid is going to identify the mid-level intensities within the image. And the level set dark is going to identify the darkest pixels within the image. Now, leveraging these level set masks, you can capture quite a bit of the detailed morphology within the bright field image. If you look at this stomatocyte down in here, you can see we're characterizing both the light and the dark morphologies within the cell type. From here, from this masking, we can create features that will then help us to characterize each of these different cell types. We also have a bright spot mask, the adaptive erode mask, and a dark spot mask. The adaptive erode mask is fairly new, and basically what it's designed to do is, in the bright field image, go in and characterize these undulations and subtle differences in the texture within the bright field image. So you can think of it as a morphology mask for bright field is essentially what it is. So some of the applications that have been leveraging these new masks are things like looking at asymmetric division. It's important for generating diverse cell types in the immune system. And in this case, we're looking at CD133 and CD34 and characterizing the relative intensity levels between 2 cells during telophase. So to begin this process, you first identify telophase cells and then use component masking to identify both of the daughter cells and then calculate the total intensities within those cells. And ideas that allows you to create a compound feature that's called the intensity ratio that compares the brightness of one cell versus the other, and allows you to set up that ratio so that you can go in and gate on cells where there's one daughter cell that's brighter for a certain surface marker than the other. And it allows you to characterize and quantify asymmetric division. Another application that has been leveraging some of the new masks is looking at micronucleus calculations. So here we're trying to measure genotoxic carcinogens. Now typically this is done with the cytokinesis block assay, which is time intensive, it's labor intensive, and essentially depends on manual scoring. So here what we've been able to do is take tens of thousands of cells that have micronuclei after treatment with different grades of radiation, and then go in and not only characterize the presence of micronuclei, but also count them. And in this lower panel here on the right, you can see using the spot count and a mask that identifies the micronucleus, we can characterize no micronuclei versus a single micronuclei versus 2 micronuclei or 3 micronuclei in that image over there to the right. Another application that we've been getting a lot of questions about is looking at circulating microparticles. There's been a number of publications that have come out looking at these tiny particles. Microparticles can be on order of 20 nanometers up to about 500 nanometers. And one of the advantages of using the image stream is that because we're using a charge-coupled device, a CCD camera, we're able to, we have very low background noise. So if you tried to do this experiment on flow cytometer, you can see that the background noise from the PMT is really high, but because we're using a electrically cooled CCD camera, our background is quite low, and that allows us to pick up these very tiny particles, especially when they're fluorescently labeled. So there's been a number of groups trying to quantify micronuclei. And some in-house work that we've done is looking at exosomes, which are also potentially on order of about 20 nanometers, and look at binding of the exosomes and then phagocytosis of these CD63 exosomes. You can see just by using the simple internalization feature and these fluorescently labeled exosomes that we can characterize membrane-bound versus phagocytosed particles. We've also developed a kit for analyzing protein aggregation. This kit is basically designed to look at and quantify the number of protein aggregates in pharmaceutical formulations. The advantage of this approach is that we can simultaneously label these cells with BODIPI, proteostat to label the protein aggregate, and cyto to label bacteria, and effectively quantify silicon oil droplets with bacterial contamination as well as look at protein aggregates. The other advantage of doing this on the image stream is that we have very good resolution. So at 60x, we're looking at a resolution of about 0.3 square microns. And that gives us really good resolution on some of the smaller particles for looking at protein aggregates, bacteria, and the silicon oil. We also have implemented a bright detail colocalization 3-way. This works just like the bright detail colocalization 2-way, but it requires a high degree of colocalization of 3 probes in order to achieve a high score. So it's a way of characterizing when 3 proteins are colocalized into a single compartment within the cell. And finally, our future goals and objectives are looking at different ways to visualize high-dimensional data sets. So 1-dimensional, 2-dimensional, and 3-dimensional data is great, but if you can leverage the thousands of features that ideas can generate to look at unique morphologies as well as fluorescent intensities, we hope to make much better characterization of different dynamic cell populations. So currently with ideas, reporting is essentially a right-click and copy plot or images to the clipboard. The software allows you to batch process the statistics table, allows you to enhance the image display and export images and combine those images with certain feature values. You can include a scale bar, we can create density plots, we can do histogram smoothing, and now you can export multiple data files to FCS format for use with third-party software. But the work that we've been doing with the Novo Software has been really great and has really facilitated some of the key things that ideas was lacking with reporting. So by exporting or by opening your data sets in FCS Express, you'll now be able to simply generate reports that have high-quality dot plots, density plots, as well as histogram overlays, and generate professional quality or publication quality reports, and do that in a high throughput and rapid manner. And with that, I'd like to turn it over to Sean, who is going to go through a brief demonstration of the FCS Express software. So, Sean.
Thanks a lot. So we're just going to take a moment here to pause. If anybody has any questions for Brian before we talk a little bit more about FCS Express, please take a moment, enter them in the questions and answers box. We will try and answer a few of them now if they come in. If not, we'll make sure to answer them at the end of the webinar. All right, great. So as As Brian had mentioned, Ideas has some great capabilities, some things that, of course, FCS Express will never be able to do. But then again, FCS Express has some really unique features and capabilities in the software that really complement ideas and ideas-based data. And we're really excited to announce this direct compatibility with the DAF and CIF files that are derived from the Amnis data sets. It's something that allows scientists to, they don't have to export kind of as an FCS file or export extra images. The DAF and CIF files that come directly off of the IDEAS software are what is going to open directly in FCS Express. And Brian had mentioned quite a few things that FCS Express can do. And really what we're going to allow you to do in the beginning is work directly between the raw data, just like you would do with FCS files, any of our kind of high quality plots and images in the software, and also access all of the images that are stored and saved with the CIF files from IDEAS software. Some of the other key points for FCS Express and some of the key features is that we have a Microsoft Office look and feel within FCS Express. And in fact, this presentation that you're looking at right now on your screen has been done in FCS Express. So it's kind of shamelessly by design. Everybody has used PowerPoint or Excel or Word at some point in their life, so it makes doing analysis in a third-party tool like ours very easy to learn. Again, as Brian has mentioned, we have lots of high-quality visualizations, plots. We can report in super high-quality presentation-ready and publication-ready reports and figures. We have the ability to work with plate-based data for high content, as well as batch processing large amounts of data or large amounts of data files. And for those of you who are in a regulated environment, FCS Express has features available to you that allow you to meet 21 CFR Part 11 compliance. Things like logging and security and audit trails that can be added on to any version of FCS Express. We also offer some unique features for IDEAS customers. Again, things like batch processing, doing histogram overlays very quickly and easily. Kind of tying into what Brian started with, we can work with cell cycle data and model that directly using the MultiCycle cell cycle analysis tool in FCS Express. We also have proliferation modeling, kinetics modeling, as well as integrated regression analysis. So what we're going to do today in FCS Express is show you how to work with the CIF and DAF files, how you can load them up and how you can work with them in some basic plots in FCS Express. We're then going to show you a few examples. We're going to go over working with cell cycle modeling in FCS Express, again, just a kind of brief example, as well as working with regression and the integrated regression analysis tools and batch processing through a series of data files. Now, if you do want to join us for a more advanced overview of the features in FCS Express, there will be a webinar directly following this webinar at 1 p.m. Eastern Time at 10 a.m. Pacific Time. It's not too late to register for that. You can simply visit the Novo Software website, click on the second webinar that's in the screen, and you can join us for that. So to get started on how do you load this data and how do you start working with it in FCS Express? Well, again, everything looks and feels like Microsoft PowerPoint. And in PowerPoint, you would start with a blank PowerPoint slide. And in FCS Express, you start with what we call a blank layout. You can see here that there's a page that appears on the screen. And this is where we're going to be inserting things like images, data analysis plots, all through our Insert tab. Again, just as you would do it in Microsoft PowerPoint, we can insert shapes, text boxes, and all the different type of analysis plots that you're used to. So to get started, what we'll do is insert a dot plot. And when you insert a dot plot in FCS Express, the first thing that it's going to ask you is to go and find the data files that you'd like to use. So what I'm going to do is open this up on my other screen and bring it over. And give me a moment here while I bring this up. And you can see here that in this file folder, I have DAF files that were acquired on the Amnis image stream. And you can see that in our files of type here now with FCS Express Image Cytometry, or FCS Express Plus, that the Amnis.DAF files are located here. You can now just choose this files of type and access that. Of course, we have every other file of type that's used for flow and image cytometry data. This is what you want to choose. You can simply click on the file and click Open. So FCS Express is going to load up your Amnis image stream data. You can move these plots around anywhere you'd like on the page very quickly and easily, just like editing objects in PowerPoint. And when you're ready to pick and choose which parameters you want to view, you simply click and hold on the axis, choose the parameter of interest, and click and hold on the next axis, and you can choose another parameter of interest. Now, one of the nice things about working in FCS Express is that you can zoom in on this data too. So if you have some parameters that are kind of squished in a corner, or you need to adjust that visualization a little bit, you can right-click on the plot and choose Magnify Resolution and quickly zoom into the parameters of interest associated with that plot. Now, one of the other nice features of working with plots in FCS Express is you can see here we have many different plot types available. Dot plots, density plots, color dot plots, contour, quite a few options here. And at any time, if you'd like to change the plot type you're working with, you can simply click on the Format tab and pick and choose what type of plot you want to look at. So if we wanted to move to a contour plot or a density style display, very quick and easy to do that and kind of see how that data changes and its visualizations for you to look at. Of course, we can also insert histograms for this data. So if we wanted to quickly insert a histogram and change over to our intensity MC11 parameter, which is our cell cycle parameter for this data set, you can see that we've generated the curve. Again, we can kind of quickly zoom in here on that data set to see what's going on. Now, you're probably used to setting up gates or thresholds in IDEA software. Of course, we can do that in FCS Express. We handle this through the gating tab. You can see that we have a number of gates that we can choose from. I'll choose a rectangle gate and insert it in here. And when we do that, you can see that the gate has been created. And one of the really nice things about FCS Express is that the statistics will update for that gate in real time. And if we apply it to another plot, all of our gating is done in real time. Now, this really comes in handy when you're working with images. So let's kind of cut to the chase here. I'm sure everybody wants to see these images working in FCS Express now. Well, we handle images from the Amnis image stream by inserting what we call a data grid. Now, when we insert a data grid onto the layout, you can see that every parameter for each cell is listed, and all the way at the end of this list are the images that are derived from the Amnis image stream. You can see that we can scroll through this list, we can make it larger, we can make it smaller to kind of see all of the cells and all of the images associated with these. Now, this may be useful to view all of these parameters, but if you don't need all these and really all you're interested in is the images, well, we can do something simple like right-clicking on this plot and choosing Format, and we can choose in our parameters to display to only view a few of the specific parameters that we're interested in. So in this case, we have some MF kappa beta staining, some DREC5, some of the bright field channels. Another kind of nice feature here is that we can show any gates that a cell falls within. So when I do this, you can now see that we're only viewing the image parameters. This data grid can be made larger, it can be made smaller to view these cells. And in FCS Express, you can even drag these cells out now onto your layout, and you can insert a nice little picture of what that cell looks like. So if you found a cell from a particular population, you want to use that as a representative image. Of course, you can take that, just kind of put it onto your plot, and now you have a nice representative image of what that particular cell looks like. Now, to go with the live updating gating, these data grids that we insert, you can simply apply a gate directly to it, and you can notice now as I update this gate that the cells that are appearing within that gate are updating for us live in the data grid. So again, the speed that's involved in FCS Express in loading in these large image-based data sets is quite fast. And again, if we create additional gates within this plot, Let's see if I can find my gate view that appeared on the other page. You notice that in our data grid, some of the events fall within the red gate, indicated by this little red box. Some of them fall within the blue gate, indicated by the blue box. And this allows us some backgating information associated with these data sets on the fly and as we work with these things. So again, working with the gates and the plots, all of this within FCS Express is quite simple. It's quite easy. You're simply working with any of the plots that you're used to working with flow cytometry data. They're super high resolution, super high quality, and you can work seamlessly between your raw data files and your images now at a click of just a few buttons. Now what we want to do is show you just a few examples of what these reports in FCS Express can look like and what you can do with them. Before I do that, I do want to stress that we also have easy to create overlays in FCS Express. I've heard that's somewhat of a pain point for people who need that currently. What we do to create histogram overlays is simply choose any of the data files that exist in what we call the data list that we want to overlay. You drag that onto a histogram plot. You choose Add it as a new overlay. And what you can see here is that an overlay has been created. Now just to kind of show you how these overlays work, they can be fully formatted. So if I wanted to say, let's fill the areas of these, put in some transparency, maybe do a little bit of smoothing. Maybe for one plot I want to look at one gate, and for another plot I want to look at a different gate, even changing the colors of these overlays. And you can see when we preview this, we have some really nice histogram overlays. If you remember, I changed the gates to display on these, and as we update the gates that we've created in our initial plot, you can see that our histogram overlays are changing. Right. So we'll show you a little bit more about that. Of course, we can view these histogram overlays in two-dimensional plots and 3D, really any way you want to visualize that. So to start with kind of some of our examples we put together for you here in FCS Express, as Brian had mentioned in the beginning of the webinar, one really nice feature in FCS Express is that we have the ability to insert what's called a MultiCycle DNA Analysis plot. This allows you live updating modeling of any cell cycle parameters that you have in your data set. You can see here that we've actually inserted a MultiCycle DNA plot. We've inserted some statistics for this plot. So we're modeling the amount of G1, G2, S phase, and all the kind of relevant cell cycle parameters associated with this. Again, all of these plots and the statistics and the calculations will update on the fly for us. So as we make a change to one gate, our model is going to be recalculated, and all those statistics for that model are going to be recalculated in real time. This also comes in handy if you want to go back and start looking at which cell cycle phases your particular cells are falling within. So you can see here that we've highlighted G1, S, and G2 cells as red, yellow, and green. We can see here the backgating information associated with these cells, which is showing us which of the populations these fall in. So really a kind of nice way that you can report this is if you want to take a look and say, all right, here's a G2 cell, well, you can simply drag this out. If it's a representative image, we can stick it in our analysis. And if we wanted to put a nice little representative image of what a G2 cell looks like into our MultiCycle analysis plot, we can do so just like that. So again, creating these reports it's very quick and easy. It's really not much more than inserting a few objects on the page and changing some of your formatting. You can see here we're just using some black backgrounds and some histogram transparency to make it look really nice. When you're ready to report this data, it's equally as easy. You can simply click on the File tab here. You can click Export, and we can export to PowerPoint to PDF. We'll do a quick PowerPoint presentation. And PowerPoint is going to launch once this analysis is done. And you can see that we are now in PowerPoint. We have a high resolution report. These plots can be blown up as large as you like. You can change the DPI, the resolution on these things. So if you're ready for a poster, if you're going to Cyto in a few weeks and you needed this, you could certainly do it there. Now, this is great for working with one data file, right? where we do a single export to PDF or to PowerPoint. But what happens when we have multiple data files? We generally don't run an experiment with just one set of data. Well, there's a few things we can do. One of the options is using what we call batch processing in FCS Express through our batch analysis tab and using templates. So as an example of that, I would like to open up here a batch analysis template that we created for a different set of data. This is going to be for an apoptosis data set that we'll open up here. And let's open up our batch analysis template. So the other thing to remember is that when you create an analysis in FCS Express, it's not a one-off thing. You can use this over and over again, so every time that you repeat this analysis, you can quickly and easily load it into FCS Express and report it. So we've created a template analysis here where we're simply going to add new DAF files to our data list. This is from our apoptosis example. So you can see here I have a series of time points for an apoptosis experiment. I click Open, and in FCS Express, all of our plots populate. I'm going to change the first overlay on this plot, and I'm also going to tell FCS Express quickly which images I want to view. So I'm just going to come in here and make sure I'm looking at our DRAC5, our Nexon5, our CastPace, and our Brightfield. Click OK. And you can see now that we've loaded in all the images and our experiment is ready to analyze. We've also inserted a text box into our analysis. We've inserted a live updating, what we call token here, for a file name. And what this will do for us is as we change between samples, you can see that our statistics, our file names, our histogram overlays, our images, everything associated with this has updated in real time depending on which file I've chosen from our data list. The other thing that we've done behind the scenes here for you is that we've already created a batch analysis and our batch actions where we've said for all the files in our data list, export to PowerPoint, to PDF, and to Excel. So what that means is once I've loaded this template and I've loaded new data here, I can simply click the Run button in the Batch Processing tab. 3 actions will be completed for each of the samples in the data list. You can see how quickly we're actually going through the data list, considering that some of these data sets are gigabytes in size for the images. And we're creating our PDF, our PowerPoint, and our Excel presentation from this. So here comes PowerPoint and Excel. So for our 6 different time points, we've exported the percentage of cells for each of the gated populations for our apoptosis experiment. We've also created a high-resolution PDF that summarizes each individual sample on separate PDF pages here. And of course, we always like exporting to PowerPoint. So here's our PowerPoint presentation. And if we needed to quickly come in and say, let's look at one of our histogram overlay slides and make it a little bit bigger, get ready to export that for some sort of laboratory notebook or presentation, we can quickly and easily do that. Now, we do understand that you're not always going to be working with a single data file at a time. For this sample data set that we have a 6-point titration series or 6-point time series for this apoptosis experiment. But there's really great ways that you can actually analyze multiple data files within the same layout and use our live updating regression curves for this. So, when you do end up with a Excel report like this, and you're looking over a time course, generally what you'll be doing is in Excel or in Prism, you'll be plotting this data, of course, against time, the percentages of cells. But we can take that a bit further in FCS Express and move away from that middleman of exporting to another software by opening up all of the data files into a standardized template. So here's our 6-point time series, time point 1, 2, 3, 4, 5, 6 going down the list. And on our second page here, we've actually calculated the regressions associated with this, something that you may be doing in Prism or Excel. What we can do now is as we adjust the gate for any of these parameters, you're going to see that our regression analysis is updating in real time. including any of our regression fits. You can see here R squared, whatever kind of regression statistics you want to put in there will update. So when you really need to kind of narrow down, maybe exclude some outlier populations, whatever you may need to do, you can actually see all of this happening on the fly in real time. There's really no need to kind of go out to a middleman for this type of reporting. It's enough to kind of work with ideas in FCS Express. We don't really need to worry about going to Prism or Excel at this point. So one of the other things I wanted to mention, a question that I did see come in about working with histogram overlays. We showed working with one histogram overlay in this kind of basic example, but when you do have many overlays, you have many samples like we do here, you can simply take all of those samples, add them to a histogram, and you can see that at that time, all of those overlays will be added to that histogram. Now, of course, we probably want to format this a little bit so that all of our overlays don't look so confusing. We'll just smooth these up a little bit. We'll fill them all and add a little bit of a transparency to it. And you can see here that our histogram overlays are looking a lot nicer at this point. If we do want to take this, in this case we have 6 different files, and we want to put this into maybe a 3-dimensional view, we can do that to see these a little bit more easily. Or better yet, one of the ways I really like looking at this in FCS Express is through what we call a stacked overlay, where we take all of the time points, we stack them on top of each other into histograms, and we can view them side by side. This also allows us the ability to set a marker on this histogram overlay and generate the statistics for all of the plots simultaneously and all of the overlays in here simultaneously. So again, kind of very quick and easy to get to the information that you need. One of the things that you can do here as well is you can see that, maybe I don't actually want to go with all my histogram overlays. Well, there's an undo feature in FCS Express where we can just click undo. We can get back to right where we started. So you can see here we can get back to right where we went, and if you need be, you can always quickly redo whatever it is you need to go to. So that function is really unlimited in the software. It's going to work with every single feature that you use in here. It's not like it's going to work only for batch or for inserting plots. Really, whatever you do, it will work for you in the software. So with that, I just wanted to summarize a little bit about what we talked with in regards to FCS Express today. Again, we're just showing you a kind of a bit of the basic functionality, and we're again very excited that we have this direct compatibility with ideas, derived, DAF, and SIF files. It's been a pleasure to work with the team at Amnis to enable this. We see that there's definitely a need in the field for working with some of the third-party tools that are out there, such as FCS Express with these data files. So we hope that people who are using ideas can keep FCS Express in mind for maybe some of the high content batch reporting, some of the kind of advanced figure generation and visualizations that you may need. And again, we have all of the kind of direct compatibility that you need working between the raw data, the plots, and the images. All this is going to come together to allow you to batch process, work with the histogram overlays, any sort of modeling that you want to do, as well as integrated regression analysis. FCS Express and Novo Software also has some very big announcements coming out next week in conjunction with the Cyto 2016 meeting. So please keep an eye out for some messages from us or possibly from the Amnis team about some new functionalities that are going to be added to FCS Express that will kind of help advance the things that you're seeing here even more. So we do have a free 30-day trial of FCS Express on the Novo Software website. Please note that this SIF and DAF file compatibility is not quite available yet. It's going to be out within the next few months on the FCS Express side of things. We will keep you updated, everybody who's joined us for the webinar today, about that compatibility and when it will be made available to everybody. But of course, you can email us at support@Novo Software with any questions you may have about that. So So I'm going to be bringing Brian back on the line here. Brian, did you want to put up a little summary about Amnis as well here?
Yeah, so if there's any questions that people have, you can contact us at Amnis support at the phone number, or contact us at the support email, and we'd be happy to talk about any of the ideas, enhancements, and compatibility with FCS Express.
Great. So again, thanks everybody for joining us today. We're going to be wrapping up this webinar in just a few minutes and opening up the second webinar for session of the day just after this. At this time, if there's any questions, feel free to ask them in the question and answers box, and we can answer them there. Thanks again for everybody.